Okay, good afternoon, everyone. Uh, First of all, thank you very much to Sanjana, the organizing team, for inviting me, particularly since I don't work on muscular dystrophy as such, and also making all the wonderful arrangements over here. What I'll do in the next several minutes is try and talk to you about some work that we have done, which speaks to a problem that several previous speakers have alluded to, and that is the need for suitable models that can be used for developing drugs and for treatment of specific disorders. I will not speak about muscular dystrophy, but what I will say applies equally uh, to those disorders. Um, Next slide, please. So what I'm going to talk about is neurodevelopmental disorders. These are defined as a condition with an onset during human development in utero, and they produce deficits in the development and structure of the brain that manifest as impairments in brain function. There are many types of neurodevelopmental disorders. Clinicians in the audience will recognize them. They're both of the sensory and the motor type. But overall, they leave the patient with some sort of deficit in normal functioning of the brain. Next slide. Uh, According to the last systematic survey, there are approximately 193 million people in India with some form of mental illness. That's about 15% of the population. And a third of these have a condition called idiopathic intellectual disability. That is to say that with varying degrees of severity, there is some deficit in brain function that may vary from just mild delay in achieving milestones through to frank mental retardation. And you can see the distribution of these cases across the various states in India. Next slide. Yeah, so that's the diseases recognized in the clinic. But there are also rare genetic diseases, such as the ones we're talking about here today, that fall into the category of neurodevelopmental disorders. And in OMIM, which is what most of us refer to, there are about 7,000 diseases where there's a single gene identified. A tenth of these affect the brain in some way, and you can see that the single largest group, shown in green on the pie chart, is neurodevelopmental, and the yellow, which is the second largest set, is the so-called mixed, which is typically a combination of epilepsy with delayed neurodevelopment. So how do we understand these diseases, and how do we find solutions for them? Next slide, please. There are a couple of fairly difficult problems to deal with. First of all, in the case of neurodevelopmental disorders, the reasons are both genetic as well as environmental, and it is often not easy to pass out these two when faced with a patient with this disorder. The second is that even with the most advanced imaging technologies available in the clinic, it is not presently possible to monitor the activity of individual cells in the brain at the level of physiology and cell biological processes. And therefore, it is not easy to watch live cells of the human brain in action and understand what is going wrong that ultimately manifests itself as the disease. So how do we get around this problem? Next slide, please. Well, I'll illustrate how we can do this by one particular disorder that we are studying. It's called Lowe syndrome. I guess it's an ultra-rare disorder. Worldwide, it's less than 1 in 100,000 or so. And it results from a mutation in gene OCRL1, which encodes for a particular enzyme that mediates the turnover of lipids in cells in the brain. There are three tissues that are affected: the eye, the kidney, and the brain. And in my lab, we focus mostly on the effects on the brain. And the phenotypes in the brain vary from mild intellectual disability through to mental retardation, seizure susceptibility, particularly in the context of fever, hypotonia. And since these patients are now living longer and longer, and the oldest one I met so far is 55 years old, there are substantial mental health issues that arise, accumulating from all the multiple problems that they face as they grow up. Next slide, please. So there are some questions that are unanswered. Is Lowe syndrome a neurodevelopmental disorder? Can we understand what goes wrong in the brain cells of such patients? And can we then correlate that back to the clinical features? Next slide, please. So to try and do this, we identified a family in Bangalore that is affected by Lowe syndrome. You can see the 3 children affected in red. And without using any particularly sophisticated technology, we simply used Sanger sequencing of all the 23 exons and identified a stop codon in exon 8 that is the founder mutation in all of these 3 patients. And this results in effectively complete loss of protein function. Next slide, please. So as you know, unlike disorders of the kidney or perhaps even blood, it's not possible to get a biopsy of the brain. And to get around this problem, what we have done is to use modern stem cell technology, which is being referred to. But for the benefit of the varying audience, this is essentially what it is. So during development, one normally goes from a fertilized egg giving a zygote through to a human. What we do here is to take blood cells or skin cells from such a patient and then reprogram them back to give you cells which are as close to the first cell from which we were all derived. And finally, taking those stem cells, we use our knowledge of brain development and in the dish convert them into the brain. And I guess in the case of muscle disorders, one would convert them into muscle. Next slide, please. And you can do this starting with a sample of blood, and along the way, you can do many things that have been referred to during the day. For example, you can biobank them, you can edit them, you can induce reporters, and finally, you can study how they are working in the dish in front of your eyes. Next slide, please. And this is an example of such stem cells. And what's shown over here is that these stem cells can give rise to all the 3 germ layers and therefore to all the tissues in the body. Next slide, please. What we do in the lab is we take these stem cells and then convert them into neuronal tissue. I won't go through the details, but over a period of about 3 months or so, one can start with such stem cells and convert them into neurons that can communicate with each other. Slide, please. And this is what it looks like. What you see on the top are such neuronal tissue that has been stained with markers that show you the various cells of the brain. And at the bottom is shown is a different cell type in the brain called glia, which are the supporting cells of the brain. And what we didn't know all this time, and we discovered for the first time when we did this, is that in patients with Lowe syndrome, there is a very dramatic upregulation of astrocytes in the brain. You can see that the red cells you see in the 3 patient lines over here, outlined in blue, are missing from the control line, which is shown on the left. And this was a very, very big surprise we had never expected. Next slide, please. You can also take these cultures and without fixing them, that is, as they are live, you can then study the function of the brain cells, activity of the brain cells that gives rise to our behavior. And if you watch the movie that is running on the top right-hand corner, you will see flickering of the fluorescence intensity that represents the activity of the individual brain cells, which ultimately comes out as behavior. And this allows you to study what's going wrong in a patient cell line compared to a normal cell line. And this is what we did. Next slide, please. And click twice. So if you see there on the right-hand side, you can see a normal patient cells, neurons, and they're firing very nicely and rapidly. And next to it on the right-hand side, LSPH002 is a patient line which hardly has any activity at all. So we now know for the first time that in Lowe syndrome patients, already during early development in utero, it is very likely that the activity of the neuronal cells is less than that in the controls. And that's probably why the behavioral manifestations is already present when the child is born. And this can be quantified. And if you press, can you, one more time? Yeah, go ahead. And you can quantify this and you can quite clearly see that the 2 patient lines have much less activity than the control. Next slide. So of course, the brain works on principle of electrical activity. And so we monitor the development of electrical activity in these cultures. And the red bars which you see on the top in the control cell line represent mature action potentials from neurons. And it's very obvious even to the lay members of the audience that in the patient lines, you can see no mature neurons at this particular age of development. So the brain is developing much slower in terms of its activity. So how and when does this happen? During development of in humans, we go all the way from stem cells, which are shown as HIPSCs, through an intermediate called neural stem cells. And those neural stem cells then form rosettes, and the rosettes elaborate to give you immature neurons and subsequently mature neurons and astrocytes. So at which point during development does the loss of the Lowe syndrome protein affect activity and brain structure? To try and understand this, we turn to a technology called the organoid technology. You heard it being referred to during Arka's talk. What we're trying to do here is to recapitulate the three-dimensional organization of the brain during development. One starts with the iPSC lines, and then over a period of time, you can see the cells growing in 3 dimensions. And in the images you're seeing over here, on day 0, you can see that it's just a very small bag of cells. But by 200 days in vitro, that is 200 days in the lab, you can see a structure which is about 4 millimeters in size. And although it looks like a ball of cotton, this is actually a mini brain growing in the lab. Next slide. And if you then section this brain organoid and look at it, you will see that the layers of the cerebral cortex, which is what gives rise to the function of the brain, are quite accurately recapitulated. You can see 3 of the 6 layers of the cerebral cortex that will ultimately form by the time the child is born. Next slide. So there has been a lot of discussion here about how to screen for drugs and whether a drug, if found, can reverse the pathology in the patient's cells. And one of the goals that we had in this project was to try and ask the question if we could at least in principle prove that this is possible. And so to do this, what we did was to take the neural stem cells from the patient lines and differentiate them to give neurons. But at the last 7 days of this culture, we have added a compound which on the basis of our knowledge we thought might be able to reverse the phenotype. And that's the inhibitor of the enzyme PIP5KC. I won't go into the details of what it is, but you can see the result very nicely on this slide. In light blue is the calcium transients from the control, and in dark blue is the calcium transients from the patient line. You can see that they are lower than in the control. But after treating with the inhibitor, you can see that the transients in the patient line have increased very dramatically. Now, the calcium transients gone up to here and are comparable. And this is the first time we've been able to find a molecule that can reverse the in vitro phenotype of the original genetic loss of the OCRL protein in Lowe syndrome. And we've been able to do this with 3 or 4 chemical variants of this compound. Next slide. A second phenotype that we tested, because you should never really go by one readout, is something else. We know that during development, the size of the brain organoids that are formed in the Lowe syndrome patient is larger than wild type. And you can see in the graph at the bottom, once again, that the blue bar is much larger than the light blue. And the dark blue bar is the patient lines. But after treatment with the inhibitor for PIP5 kinase, you can see that the size of the organoids is substantially reduced back towards wild type. So using this sort of in vitro experimental system using patient-derived lines, I've shown you that a chemical inhibitor, if you can identify it or screen for it, is able to reverse the cellular phenotypes that the patient lines show. Next slide. Okay, so Alok briefly referred to this in the morning, but when you work with cell lines and cell culture systems like this, there's really no guarantee that what you see in the cell line is what is seen in the patient's brain if you were to be able to have a look at it. And so what we did was to try and ask whether we could correlate our results with what is already known from clinical analysis in patients. And on the left-hand side over there, you can see MRI scans from children with Lowe syndrome, sex-linked recessive, so these are boys. And what you will see, the arrow marks on the MRI scans, is the presence of these hyperechoic structures, which are very typical of Lowe syndrome patients and which increase as a function of age as the child grows older. So a colleague of mine some years ago had tried to do a comparative analysis of this in zebrafish, for which he made a zebrafish knockout of OCRL1. And what you can see over here is an MRI scan of the head of a zebrafish. And here you will see in the boxes that are shown over there that very similar hypoechoic lesions as seen in the patient MRI scans are seen in the mutant zebrafishes at the bottom compared to the wild type zebrafish, which are at the top. And if you take these zebrafish heads and stain them and section them, you will find that there are enhanced acidic staining in the OCRL1 minus zebrafish brains that is quite similar to what you see when you stain for astrocytes. In other words, what we found in the cell lines, which is that there's an increase in astrocyte numbers, is recapitulated in the zebrafish knockout in the brain and is very likely similar to what is seen in the patient MRIs from the children with Lowe syndrome. Next slide. Okay, so I was apparently supposed to speak about gene therapy. I think Chandak, in his enthusiasm, put down the title as Experiences from Gene Therapy. But I have never really done any gene therapy. So I thought I would just reflect on a couple of points on the basis of my thinking about this for some time. So when you have any human disease that arises from a particular mutation, you end up with a patient where the tissue in the patient is affected in some way. And one has to stop for a moment and reflect and ask the question, why does that happen? Does it happen because the gene that you're studying is required in the adult cells of the patient? Or is it because that gene is required during the development of that tissue and is then manifest as clinical phenotypes in the child or the adult? This is a really important question to answer. So for example, is dystrophin required in adult muscle cells after they've been fully formed, or is it required during the development of muscles? And what you're seeing in the patient is actually because of a defect in development. This is really quite important. You've heard about the advances in ways of delivering genes that are being developed. But once they have been developed, one is going to be faced with a very important question. At what point do you correct the defect? Do you correct it in the adult patient? You try to correct it in utero, you try to correct it just after birth. So this question is a very important one, and it'll come up over and over again. The second point is that many tissues are not composed of one cell type, and there are more than one cell type. And one will then have to reflect on the point as to which cell type are you going to restore it to. In the example that I've just shown you, are you going to restore it to astrocytes? Are you going to restore it to neurons, or both? So these are very important questions to think about. Final slide. Okay, so as Arka just mentioned in his seminar, it's going to take some time to get to the point of being able to readily do gene therapy. But what do you do for patients in the meantime? Well, one way that you could think about this is, first of all, you need to understand the mechanism of the disease. And then you could try and rewire the cellular biochemistry using small molecules, using either in vitro or ex vivo human disease models. And what I've shown you today in my talk is some examples of those kinds of models. And that will go a long way towards shortening treatment developments. And you can do that quite effectively now. I've just shown you an example with one compound that I worked with, but we have available more than 50,000 compounds, most of which have passed either FDA, EMA, or other agency approvals. And what that means is that these 50,000 compounds have now been pre-selected for studies of safety and also of bioavailability. These libraries are available commercially in India and at relatively low cost. And it is possible to do, for example, a high throughput screen using the type of assay and cell model that are used to try and find new targets that one can then take to the patient with a relatively short time lag. Next slide, please. The final one. And so I'll end by just thanking the family members of the two families that we worked with. A large number of people in my lab contributed to developing this project. And my clinical colleague and friend, Dr. Anil Vasudevan, who looks after the patients and makes sure that they're okay. And the last slide. And we would not have been able to do this without sustained funding support from many different agencies. both government as well as philanthropy. So I'll stop here, and if there are questions later on, I'll be happy to answer them. Thank you.
Okay, thank you, Dr. Raghu.